whiteboard?
Yes, sir.
Yes, sir.
Okay. So we first talk about the protein folding. So what exactly happens? Like uh, this is the say ribosome. Now when the mRNA, this is the mRNA, okay, passing through the ribosome, and let us say that this is the protein that is coming out. Okay, so once the protein comes out of the ribosome, now immediately what happens? This protein it undergoes folding, something like this. So this is what is the folded structure of the protein. Okay, this is what we call protein folding. So that means eventually what happens after coming out of the ribosome, this protein it folds immediately. Okay, so now the question is that what type of structure the protein will assume. That depends. Say that depends upon the amino acid sequence of the protein. Okay. So what happens? This protein it folds in many different types of structures. Okay. But ultimately it will assume that structure of the protein which is having the lowest energy. Okay, because as per the second law of thermodynamics, uh, all the molecules they try to remain in the lowest possible energy. Okay, so that is why only that structure of the protein ultimately the protein will assume when which is having the lowest energy as per the second law of thermodynamics. Second thing is that the protein assumes that structure such that the hydrophobic amino acids they are inside the protein, they are not exposed outside, so they're always inside the protein. This is how the protein is to fold. Okay, now this regarding the protein folding. Okay, the question I gave you in the WhatsApp group that once the protein is still it is being synthesized in the ribosome. Okay, so that time itself the protein, okay, let us discuss about the protein misfolding. Now, what is protein misfolding? Sometimes what happens in the event of folding, some proteins they fold in the wrong manner. Okay, so the wrong folding can occur because of the variety of reasons. So one of the reason is the stress. So if you give the stress to the cell, so what happens many a times the proteins they fold in the wrong manner. The wrong folding occurs, then what happens? These proteins they undergo aggregation. So what is wrong folding now? Wrong folding means I've already said that all the hydrophobic amino acids they should be inside the protein, and the hydrophilic amino acids or polar amino acids should be outside. So hydrophobic or non-polar amino acids should be inside. So if I write it here, I would say that all the hydrophobic or non-polar amino acids should be inside the protein, and all the polar amino acids should be exposed to surface of the protein. Now what happens when the proteins fold in a wrong manner? So in that case, what happens? Supposing that so this is the protein okay, which is being synthesized. Now let us assume that this particular part of the protein, say, I would say this part of the protein, okay, it is comprised of, suppose, hydrophobic amino acids. So in that case, this particular protein, it should fold in this manner. So that means this particular hydrophobic part of the protein should be inside. Now, on the other hand, if the protein folds in wrong manner, so in that case, what will happen? Say, this is the wrong folded protein. So here, what is happening? This particular portion, say this particular portion of the protein that is made up of hydrophobic amino acids, it should be inside, but now it is exposed outside. So this is misfolded protein. When the misfolding of the protein occurs, so what happens? Many proteins, many of the misfolded proteins, they will aggregate. How they are going to aggregate? So let me show you this manner. Say, for example, this is one protein, okay, and this part of the protein is made up of hydrophobic amino acids. So this is another protein, same misfolded. It is not folded. This part is hydrophobic. This is another protein molecule, same protein, but another molecule. Okay. So now what will happen once this kind of unfolded or misfolded proteins are present? So what will happen? They will undergo aggregation. So how they are going to aggregate? So this hydrophobic parts, they will combine together, because the hydrophobic parts in the aqueous solution, they always come, because the one which we call hydrophobic interaction, so in the water, the hydrophobic surfaces, they try to cover each other. Okay, so this is the protein, and this part is the hydrophobic surface, so they will try to cover each other. This is called aggregation. So now in this case, what will happen? All of these protein molecules, okay, they will just join together through their hydrophobic parts. Join like this. Okay. So this is what is called protein aggregation. Protein aggregation. So protein aggregation, the proteins, they aggregate through their hydrophobic part. Okay. Now, what is the problem with this kind of protein aggregates? Okay. So in the microscope, you will find them as inclusion bodies. I think you know about in case of some viral infection, the protein aggregates, if you find some kind of cellular inclusion bodies inside the cell. So these inclusion bodies are nothing but they are aggregates of the protein. Okay, so this kind of protein aggregates are dangerous for the cell. Okay, so now the cell will have to prevent the aggregation of the proteins. Okay, so in the WhatsApp group, I asked one question that when the protein is being synthesized, this is the protein that is being synthesized in the ribosome. So during synthesis, the protein is not in the folded form, it is unfolded form. So in that case, why the protein aggregation does not occur? Why the protein aggregation does not occur when the protein is still being synthesized in the ribosome? Because it should occur, because this protein is open. After synthesis, only it folds such a way that the hydrophobic amino acids are inside. But here it is not. Here this portion is, suppose, hydrophobic amino acids, it is exposed. Okay. So here what happens when once the protein is still in synthesis, okay, that time what happens? This kind of unfolded protein, okay, in the ribosome, it is bound by one type of protein, that is called chaperone proteins. Okay, they are called chaperone proteins. Okay, so there are various chaperone proteins. So important among them are HSPs, heat shock proteins. So now there are various heat shock proteins based on their molecular weights like HSP 60, HSP 70, HSP 30. There are many. Okay. Now in this case, what happens? These chaperone proteins, what is their function? So chaperone proteins, they bind, they get attached to the unfolded proteins. Okay. And then they prevent the aggregation. So what will happen? In this case, once the protein is still in synthesis, so the in that case, the HSP 70. So this is HSP 70. So HSP 70 will go and bind the protein. So that's how what is going to happen. This HSP 70, it will go and bind the protein. It will coat the protein in this manner. So this is how these chaperones or HSP 70 proteins specifically, so they will prevent the aggregation. That is the reason why the proteins that are still in synthesis in the ribosome do not aggregate. Okay. So once the protein synthesis is over, then these proteins they will fold immediately. Just in a few seconds, the protein will fold. So after synthesis, these HSPs they will be removed. So after your synthesis is over, then this HSP they will be removed. Okay. So all of the HSPs will be removed, and then the protein will undergo folding. Okay. So HSP 70 okay will prevent the aggregation of the protein. Okay. Fine. Now this is about once the protein has it is still in synthesis, it will be bound HSP 70, and then when the protein folds, this HSP 70s are removed. Okay. Now second thing comes. Sometimes what happens? These HSP, these proteins, after their synthesis, they go inside the endoplasmic reticulum because these ribosomes they are present on the surface of the endoplasmic reticulum where the proteins they synthesize and then they enter inside the ribosome. Okay. So in the ribosome, again proper folding of the protein occurs. Okay. Now again inside the ribosome also sometimes what happens the proteins do not fold properly. There is a misfolding of the proteins that occur inside the endoplasmic reticulum. So that means proteins have folded, but they have folded in the wrong manner. Okay. So in that case, wrong manner means their hydrophobic amino acids are exposed outside. That should not be. So if they're exposed outside, if nothing is done, what will happen? All those kind of misfolded proteins, they will aggregate. If they aggregate, then that will be dangerous for the cell. So in that case, to prevent the aggregation, the second type of chaperone protein that comes into play. This is called HSP 60. So HSP 60 will come into play, okay, once the proteins are misfolded inside the endoplasmic reticulum. So HSP 60, how does it look? I'll try to draw it as it is drawn in the book. Hmm. So, so HSP 60, it looks like this. Hmm. So now what happens? The Supposing that you have a protein that is incorrectly folded. So this is the protein that is incorrectly folded. Now, incorrectly folded protein will enter into the HSP 60. Okay. So once it goes inside, now this protein will be inside. Okay. And then there is a cap present on the surface of this HSP 60. Okay. So, so this cap will close. So this cap will close. Okay. And then once the cap is closed, okay, now this will allow this, that means the protein present inside the HSP 60, it will be allowed to refold. Okay, fine. So then after refolding, once the protein is refolded and it is folded into proper structure. So let us assume that this is the proper folding of the protein. Once it is properly folded, okay, and then thereafter, what will happen? This particular cap will be removed and the protein will go out. Okay, so the HSP 60 mediated correction of the protein folding is the last attempt, okay, by the system to correct the protein misfolding. Okay, but this system also, it is not 100% accurate. Okay, now this particular proper folding of the protein, this is energy dependent process. Okay, energy dependent process means because the correct folding of the protein, because refolding of the protein requires energy, and this energy comes from ATP. So this HSP 60, it has got ATPase activity. ATPase means the enzyme that breaks the ATP. So it is having ATPase like activity. It will break the ATP, it will get the energy from that, and that energy will be used for correct folding of the protein and will go out. Now, even after, suppose the HSP 60 mediated correction of the protein, if it fails, it may be possible that even after that, the protein structure remains like this. It remains in the incorrectly folded form. In that case, the last option is, the last thing the system will do, it will degrade the protein. This protein should undergo degradation. So what will happen? Because in this particular protein, okay, the hydrophobic amino acids are exposed outside. So what will happen? So when the hydrophobic amino acids are exposed outside, so what is going to happen? Says this portion is having the hydrophobic amino acids. Let us assume like that. Okay. So what will happen? There is a another type of protein that is called ubiquitin. Ubiquitin. So ubiquitin is a small protein. Okay. So this ubiquitin protein, it is something like, it is a tag. The tag that tells the system that this is a waste protein. Something like this, you have a dustbin, okay, you have a garbage, so the garbage just put the label. So the person knows that, okay, this is the garbage to be put into the dustbin. So ubiquitin is just like a label. So what happens? Say this is the ubiquitin molecule. Okay, so this particular ubiquitin molecule, with the help of one enzyme that is called ubiquitin ligase. So with the help of ubiquitin ligase, this ubiquitin protein, this will be placed into the amino acids which are hydrophobic. That means the ubiquitin has got affinity for the hydrophobic amino acids. Only those hydrophobic amino acids which are exposed outside. So in this example, so here the amino acids are hydrophobic amino acids are exposed outside. So ubiquitin ligase will put this ubiquitin over here. Okay, and then this ligase, it will make a chain of ubiquitin. Something like this will make a polymer of the ubiquitin. Okay, so this ubiquitin, okay, once forms a ubiquitin chain layer polymer over here. Now this complete protein, this will move to a kind of uh, protein, another protein that is called proteasome. Proteasome. So proteasome, it is a cylindrical structure. It is also a protein. So it is having a cylindrical structure. So so the proteasome, it is a having the cylindrical structure. So the proteasome is having a this type of cylindrical structure. This is what is the proteasome. Okay, just make it small. Okay, so this particular proteasome, it has got a cap-like structure. So one cap-like structure is present over here. Okay, fine. So this proteasome structure, it is called 20S protein, and this is the cap is called 19S protein. Okay. So now this particular ubiquitin or ubiquitinated protein, now it enters the proteasome. Okay, now how it will enter? So only those proteins can enter the proteasome that has got the ubiquitin. So without ubiquitin, it will not enter the proteasome. Once it enters the proteasome, so here it has got this proteasome, it has protease like activity. Protease means what it will do? It will break the protein into amino acids. So then after that, after breakage of the protein, then their respective amino acids will come out. Protein, After breakage, it will come out. Fine. Now, just nutshell, I will tell again. Say, once you have a ribosome, the ribosome, the protein synthesis is still going on. So the protein is unfolded. So unfolded protein has got more propensity to cause the aggregation because the unfolded proteins, their hydrophobic amino acids are exposed. So these unfolded proteins have got higher propensity to aggregate. So to prevent the aggregation of the protein, when the protein is unfolded form in the ribosome, so the HSP70 it goes and bind the protein. It makes the coating of the protein so that the protein does not undergo aggregation. Then after coming out of the ribosome, this protein will fold. Once it folds, then the HSP70 is removed. Now the folding mainly occurs inside the endoplasmic reticulum. Now inside the endoplasmic reticulum, many times the folding does not occur properly. So if you have 100 proteins, okay, say 20 proteins will fold in wrong manner. Now, there are many factors that affects the wrong folding of the protein. It may be stress, it may be a viral infection or anything. That is why in the viral infection, you find inclusion bodies. So inclusion bodies are nothing but these are the aggregates of the proteins, which occurs because of the protein misfolding. So if the protein misfolds, then what happens? They tend to aggregate. Again, in the physiological condition, what happens? This kind of aggregation is prevented, okay, by putting the protein into the misfolded proteins into the HSP60. HSP60 is a cylindrical structure. This kind of structure. So this will enter and then the cap will close. After that, inside the HSP60, this protein will unfold. Again, it will refold. Okay. So once it is refolded, so refolding requires energy. So this HSP60 has got ATPase activity. That with the help of ATP breakdown, it will get the energy to refold the protein in proper way. Once the protein is refolded properly, then it will open and protein will go out. Only properly folded protein will go out. But this system is also not foolproof. Sometimes, even if it opens, protein comes out, and you find that protein is still not properly folded. So the last option is to degrade that protein. So HSP60 is the last attempt by the cell, okay, to properly fold the misfolded protein. If it fails, then nothing. So this misfolded protein, now it is dangerous for the cell because it will form the aggregation. So to prevent that, last option is the ubiquitin is a small protein that will get attached to the misfolded protein. So where it will get attached, this ubiquitin will be attached to the hydrophobic amino acids, exposed hydrophobic amino acids. So misfolded protein means its hydrophobic amino acids are exposed outside. So the ubiquitin will go and get attached to the hydrophobic amino acids, exposed hydrophobic amino acids of the protein. Okay, and this putting of the ubiquitin into the hydrophobic amino acids of the protein is done or is catalyzed by one enzyme that is called ubiquitin ligase. So this ligase will put the ubiquitin into the hydrophobic amino acids of the protein, exposed hydrophobic amino acids, and that will form a chain of the ubiquitin. It will form a polymer of the ubiquitin. And then this kind of protein is called ubiquitinated protein. Let me write it properly here. So this kind of protein, it is called ubiquitinated protein. It's difficult to pronounce. Okay. So this kind of ubiquitinated protein, now it will move to proteasome. So proteasome, it is an enzyme that contains proteases. So proteases have the capacity to break the peptide bonds. So the protein is made up of amino acids. So and these amino acids are joined by peptide bonds. If you break the peptide bonds, all the amino acids will separate. So now this protein containing the ubiquitin will enter the proteasome. So proteasomes, they are cylindrical structures, and then they have a cap-like structure. This cap will allow the entry of that protein only which has got ubiquitin. The protein without ubiquitin will not enter, and then the protein will enter the proteasome. And inside the proteasome, you have the protease enzymes. These enzymes will break the protein into respective amino acids. Then these amino acids will be released to the cytoplasm. Okay, so this is how our system first tries to correct the misfolding. Okay, and then ultimately, if it fails, then the only option is it will degrade the protein, and degradation occurs in the proteasome. Okay, now we'll discuss about there is a protein called prion proteins. So you know that there is a disease called mad cow disease, CJD, Creutzfeldt Jacobs disease. Okay, then crazy ship disease, all these things. So now, what is the causative agent of this disease? It is the prion bodies. So what are the prion bodies? So prion bodies, these are the membrane protein. That means prion proteins. So prion proteins, okay, so they are present on the plasma membrane of neurons. And the exact function of the prion proteins is not known. Okay, but when they become now these proteins, okay, when they are properly folded, there is no problem. But the problem comes when they are improperly folded. Okay, say for example, this is the cell surface, neuron cell surface. So in the cell surface, say this is the normal prion protein. Okay, fine, it's present. Okay, but then this particular prion protein, okay, its structure gets altered, or the misfolding of the prion protein occurs. When the misfolding of the prion protein occurs, then it causes the disease. Okay, so how this misfolding occurs? So it is believed that this kind of protein, the prion proteins, the misfolding generally does not occur in normal condition. Even if you try to misfold the protein, it will not occur. So it is very much robust protein. Okay, so this particular protein misfolding generally does not occur. Okay, but how the misfolding occurs in case of disease? So in case of disease, say for example, this particular protein is the misfolded protein. Okay, now what happens when this misfolded protein comes in contact with the properly folded protein? When it comes in contact with the properly folded protein, so what happens? This properly folded protein also, it becomes misfolded. So that means in this case, the protein misfolding is a chain reaction. So in normal condition, okay, just like I have talked about the protein, they misfolding occurs in many proteins, but in prion proteins, misfolding generally does not occur. Its sequence is like that. It's very, very difficult to cause the misfolding in the prion proteins. Very difficult. But how the misfolding occurs? These prion proteins, Okay, if they come in contact with another plant protein which is misfolded, then immediately this will also misfold. Once it gets misfolded, so what will happen? This will interact with the normal plant proteins and it will make all the plant proteins a misfolded protein. Okay, so the plant proteins becomes misfolded. So misfolded plant proteins, they are also called plant bodies. So the plant proteins, normal plant proteins, they become plant bodies. So then what happens? All these misfolded plant proteins, they aggregate in the membrane. When they aggregate in the membrane, then they hamper the normal activity of the cell. Okay. So this is the reason how these plant bodies they cause the disease, because plant bodies are said to be infective, but they don't have DNA. They are not live organisms. Okay, they are simply the protein molecules having no DNA. But these are abnormal proteins. But the question is how one abnormally misfolded protein can cause misfolding of another protein. It is still not understood. The exact mechanism is not understood. Okay, but only what is understood is what happens if the misfolding occurs. Okay, there will be aggregation, just like earlier.